This video explains the section of RNA for the biology syllabus. So we've already spoken about DNA, but if we look more at RNA, RNA is ribonucleic acid. So where DNA is deoxyribonucleic acid, which means that the sugars in DNA are deoxyribose sugars. When we look at RNA, the sugars are now ribose sugar. RNA molecules are a lot smaller than DNA molecules, but they're still polymers made of nucleotides. One of the big differences, though, is that they're only single-stranded, not double-stranded. And as we said, they're a lot smaller. They're usually only a few hundred nucleotides, but DNA is around 3 billion nucleotides per chain. RNA nucleotides are a lot more short-term, but DNA is store information permanently throughout the cell's life. An RNA nucleotide contains a ribose sugar, a phosphate group, and the nitrogen-containing base. So they have the same nitrogen-containing bases as DNA, except for the fact that DNA has thymine, while RNA has uracil. RNA is formed in the nucleolus, inside the nucleus, inside the cell. And RNA also has a 3' and a 5' end, which we spoke about during the DNA video. Obviously, RNA doesn't have hydrogen bonds between nitrogenous bases because RNA is single-stranded, except for a tRNA, which we'll talk about a bit later, because a tRNA, the strand is folded back on itself a little bit. So the structure of RNA nucleotide is almost identical to that of a DNA nucleotide, except this is now a ribose sugar. So it has basically an extra O, oxygen molecule, as opposed to the deoxy, so one less oxygen molecule. There's 3 types of RNA molecules. There's ribosomal RNA, which forms a part of the ribosome, together with ribosomal proteins, and this is produced in the nucleolus. And then the ribosomes are released out of the nucleolus, and protein synthesis occurs primarily, or a lot of the time, in the ribosomes, or on the ribosomes. The second type of RNA is mRNA, so messenger RNA, which carries a message for protein synthesis from the nucleus, from the DNA strand to the ribosome, where translation then occurs. Transfer RNA is probably the most important one. It transfers specific amino acids from to the ribosome for protein synthesis to occur. And transfer RNA is found in the cytoplasm. The structure of it is almost like a clover shape, where the nucleotides fold back on itself due to the attractive forces between the complementary bases. And here you can see hydrogen bonds will form in the folded back places, and hydrogen bonds hold it in place. Each tRNA molecule has a binding site, which binds to a specific amino acid on one end, and then on the opposite end, it has a sequence of 3 specific three specific nucleotides, which form something called an anticodon. The nucleotides in the anticodon, they determine which amino acid bonds to the tRNA molecule at the binding site. So if we just compare DNA and RNA, DNA is located within the nucleus, within the mitochondria, in mitochondrial DNA, and in plants it's also located in the chloroplast. RNA is located in the nucleus, and then within the nucleus in the nucleolus, in the cytoplasm, and in the ribosomes. DNA is double-stranded and a lot longer, whereas RNA is single-stranded and quite a bit shorter. DNA has a deoxyribose sugar, RNA has ribose sugar. DNA has ATCG as nitrogen bases, whereas RNA has AUCG, so uracil instead of thymine. The functions of DNA are, within DNA there's genes, within a chromosome there's genes which code for proteins, and DNA also contains hereditary information. RNA, its function is protein synthesis. So if we look at protein synthesis, there's 2 stages. There's transcription and there's translation. Transcription comes first. You can remember it simply because transcription comes first in the alphabet before translation, encryption before relation. In transcription, what happens is RNA is produced in the form of mRNA, and in translation, this DNA code of a gene is transformed into the actual protein. So the amino acids link up, form the protein chain. So the transcription, it's also quite a good flow diagram question, but it's quite long, so they'll sometimes just ask you for a part of it. Sometimes they'll ask you just for the whole of transcription and not translation, but it's a good idea to learn this process quite well. So DNA acts as a template strand for the RNA synthesis. We saw during DNA replication, the two strands of DNA separated, and they each acted as template strands to produce more DNA molecules. Now DNA acts as a template strand to produce mRNA. So the hydrogen bonds between a portion of DNA break. Keep in mind that during DNA replication, the whole DNA strands, the whole chromosome separates. But for protein synthesis, only a portion of DNA breaks, so it would be only at a specific gene. The genes code for proteins. The specific arrangement of nitrogen bases is what acts as the code for the proteins. The parts of DNA that aren't coding for proteins so they aren't genes, are called junk DNA, but this name's kind of misleading. Scientists have found that they're not junk. They just have a different purpose as opposed to genes. So once the hydrogen bonds are broken and the portions are separated, free Rna nucleotides in the nucleoplasm position themselves opposite the complementary complementary nucleotides, and the hydrogen bonds form between them. So the A matches with the adenine of RNA matched with the thymine of DNA, uracil of RNA with adenine of DNA, C with G, G with C. Then the ribose sugar of one nucleotide, so from the free nucleotides, bonds with the phosphate group of the adjacent nucleotide, which forms the sugar phosphate backbone. This process of RNA production is catalyzed by RNA polymerase, the enzyme. But once the whole RNA strand is complete and the backbone is formed, it breaks away from the DNA strand and it moves to the ribosome in the cytoplasm, so out of the nucleus, out of the nuclear pores, to be used for protein synthesis. Then the two DNA strands reform, the hydrogen bonds reform, and the DNA rezips up. This is all mRNA that we formed here. So there's a site on the DNA at the beginning of the gene called the promoter sequence, or called the start signal, which tells RNA polymerase this is where you must start lining up the free RNA nucleotides. So from the start signal onwards is where free RNA nucleotides line up, and then they end at the stop signal or the terminal signal. That's what RNA polymerase uses. The gene that's being transcribed is called an open reading frame. I think I have a picture here. This would be the open reading frame, the start signal, the terminate signal, and the promoter gene. There's a few genes in before the start of the gene, between the promoter and the start of the gene, just almost for the RNA enzyme to get ready. So the DNA strand that's not being transcribed, when the DNA splits open, we know it's double-stranded. The top strand, for example, one of the strands would be transcribed, and the RNA would join to it and then form the mRNA. The bottom strand is called the non-coding strand or the complementary strand. It could be a gene used to code for a different protein, or it could be a section of junk DNA. So this would be how the process occurs, and you can see it only splits in a certain portion, not the entire chromosome splitting. mRNA moves out of the nuclear pore into the cytoplasm. So what are proteins? Proteins form a large part of the ground plasm of cells, so just the building blocks of cells. A lot of proteins are enzymes to catalyze and control chemical reactions. They're found in membranes in and around the cell, and some hormones are proteins. Proteins form part of the hair, nails, hooves, feathers, so a lot of important parts of organisms' bodies. And proteins are made of monomers called amino acids. So we saw earlier that the monomers of DNA are called nucleotides. Now the monomers of proteins are called amino acids. There are 20 different amino acids, and obviously there are many different proteins because of different combinations of amino acids. We see they differ here in how many amino acids in the chain, the arrangement, the sequence of amino acids in the chain, which type of amino acids in the chain. So you could only use 3 amino acids of the 20. You could use all 20. You could use a different 3. That also differs the protein. The way that the protein is folded and the cross-linkage, so the structure of the protein, and the number of polypeptide chains. And polypeptide chains we see here are a long chain of amino acid. So 3 to 50 amino acids is a polypeptide chain. More than 50 amino acids is a protein. DNA controls the cell by controlling what proteins are made. So DNA controls which codes are transcribed into RNA, and since these codes that are transcribed into RNA are then translated into a protein. That's how DNA controls it. So as I said, this is the structure, or the basic structure of a gene. A gene is made of double-stranded DNA, and it has the promoter, the start site, terminator site, and the open reading frame in between. So the second part of protein synthesis, we've done transcription, now we look at translation. So as we said, messenger RNA was formed during transcription. Along the messenger RNA molecule, there's a sequence of nucleotides, but they're read in blocks of 3. And each block of 3 is called a codon. We spoke earlier about the anticodons on tRNA. So at the opposite end to the amino acid of the tRNA, we have a section of 3 nitrogenous bases called the anticodon. Now on the mRNA, we have a section of 3 bases called a codon. So each codon is complementary to an anticodon. So for example, if the codon sequence was AAG, the anticodon sequence would be CC. Sorry, if the mRNA codon sequence was AAG, the anticodon sequence would be UUC. And it's U obviously because we're talking about RNA. And so each codon codes for an amino acid because each codon codes for an anticodon, and the anticodon is specific to the amino acid. So the ribosome moves along the mRNA strand from 5' to 3', and it reads the codon. And in doing so, it tells the tRNA to bring a specific amino acid to the ribosome because of the anticodon that's complementary to the codon. So the anticodon binds with the codon, forming the hydrogen bonds. Now the tRNA has bound to the mRNA. And the next tRNA comes along with the next 3 nitrogenous bases, bound to the next codon, and then we have 2 amino acids lined up next to each other. Then the amino acids of these tRNAs are joined by a peptide bond. Once the amino acid has joined to the next one by a peptide bond, the tRNA is released and moves away from the ribosome, leaving the amino acid joined to the previous one. This continues until the stop codon is reached. So there's a start codon and there's a stop codon. And these start and stop codons don't code for tRNA. So they don't have a amino acid brought at that point. So the protein or the polypeptide chain, depending on how long it is, how many amino acids, when it's finished, when it's reached the stop codon, it breaks away from the mRNA. We can reuse the mRNA, and this protein folds and performs its function. So when we look at codons, there are 3 bases per codon. The reason that there's 3 bases and not, for example, 1 base or 2 base is because if there was only 1 base, then they would only be able to form 4 different codons. An A code, A, C, G, T, AUCG, adenine, uracil, cytosine, and guanine. If there were 2 codons, there'd only be 16 different combinations of anticodons that we could create because of the combinations of nitrogenous bases together. And we know that 16 is not enough because there's 20 amino acids, so there needs to be at least 20 codons to be made. Therefore, there's 3. 3 is the minimum number. And 4 cubed is 64, which is enough. And we can see that there's 64 different types of codons, but there's only 20 different amino acids. This is actually quite useful because that means that multiple codons, and therefore multiple anticodons, can code for the same amino acid. This means that the code is redundant, i.e. more than 1 codon or combination of 3 bases for each amino acid. It reduces the chances of mistakes due to mutations, which we'll look at here. And because the amino acids have more than 1 codon to code for it, except methionine, which is the start codon. That's the only codon that has only 1 combination. That's the only amino acid that has only 1 combination of bases to code for it. So there's 2 different almost diagrams. This one's quite commonly used in exams. And the way to read it, you just have to understand it. Obviously, you don't have to know that CUC codes for leucine. You just have to know that if they gave you CUC, you'd know that it's leucine. You must just be careful because they say the codon. This is the combination of bases on the codon and not on the anticodon. So if you look at it, you can look at the first base of codon, second base of codon, third base of codon. But the easier way to do it would just be to look for the specific combination. So let's say, for example, they gave you the codon GGU. Now we've got to go find which protein is coded for from GGU. Let's find it. There we go, GGU. We know that glycine is formed. If they try to trick you and they said, hey, you've got an anticodon of GGU, don't be fooled into looking and finding GGU glycine because you're looking for the anticodon. Now we need to work out what the codon is. If the anticodon is GGU, then the codon would be CCA. We look for CCA, here's CCA, proline. That's the amino acid that will be formed. This one's, again, quite easy to use. It's also the codon. And let's say they gave you a codon of GGU, you'd go GGU, glycine. We can see it's the same here. If they gave you an anticodon of GGU, you now got to find the codon. So CCA, we go CCA, proline. So you read from the inside to the outside in this wheel diagram. And as you can see here, methionine or MET is the only one with one combination. You see it here, and you see that it goes AUG is the combination for the start codon. So a gene expression, so when a gene is turned on, which we spoke about in the DNA video, it means that it's allowing proteins to be made from it. So it's exposed, this gene is exposed, which allows mRNA to form on the gene, move to the ribosome, tRNA brings amino acids, and it forms a protein. So it can be transcripted and translated. The last thing to look at in terms of RNA would be mutations. So if DNA is damaged by chemicals, by radiation or something, then changes can be introduced to the genome, into either the DNA or the RNA. This is because if a mutation happens and it causes the nitrogenous bases to change, then it may code for a different protein, or, well, a different amino acid, and then forming a different protein. Any factor that causes a mutation or that increases the rate of a mutation would be called a mutagen. Example would be chemicals, radiation, smoking, X-rays, that sort of thing. So smoking is a mutagen because it damages the DNA and it may cause it damages the genes that protect against cancer, may cause them to mutate, change their bases, and then they code for different proteins, possibly dangerous proteins, cancerous proteins. Physical mutagens are something like ionizing radiation. So there's a few different types of mutations. The two main almost umbrella terms for mutations would be chromosomal mutations and point mutations. Chromosomal mutations are mutations that affect large chunks of DNA, so like a whole chromosome or a whole section of a chromosome, and they usually occur during cell division. So Down syndrome would be a chromosomal mutation because it's a whole extra chromosome. Whereas a point mutation is very small mutations such as a single base change or a single nucleotide change. And they mostly occur during DNA replication as opposed to cell division. There's a few types within point mutation. There's a few types of point mutation. Insertion, deletion, and substitution. These are quite self-explanatory. Insertion is when a nucleotide or a base is added in. Deletion is when bases are removed. And substitution is when bases are swapped. So just the wrong base comes in. So deletion and insertion are called frame shift mutations because not only do they change this base, but they'll change all the bases afterwards as well. So if we look here, an insertion example, we've got TAC, GGC, TCA. I've split them into three because that's the codon split into three, and then the amino acid, one amino acid per three bases. So we start with TAC, GGC, TCA. Now we go TAC, AGG, CTC. So we can see that this A has been added in here. Now the A comes as the first base, and it goes GG. And then the C shifts over to the next codon. So that you can see that it affects all the bases, not only this base where the A is added, but all the bases afterwards. Whereas if you looked at a substitution mutation, for example, we'd have TAC, GGC, TCA. And let's say we were doing substitution, we'd go TAC, AGC, TCA. So this next codon would stay the same, but this codon would be the only one changed. Then you can see that it affects the RNA that's coded for, and therefore affects the amino acid, where originally we had MET, PRO, SER, we now have met, site, glue. So all the following ones are wrong. That's what a frame shift mutation is. What I said earlier about redundant mutations, the redundancy of codons, I'll talk about now. So within point mutations, we obviously had insertion, deletion, and substitution. Those are different types of mutations. But we can also look at the effects of the mutations and divide those into the types. Firstly, we can look at a sense mutation, where a new codon is formed because of insertion, deletion, or substitution, but now it still codes for the same amino acid because of the redundancy of a code. So for example, if we looked at this table, if your codon formed was originally CCU, and now we have a mutation, a point mutation of substitution, where the U is swapped for the C, so we get CCC. Lucky for us, that's a sense mutation because they both code for proline, so they both code for the same amino acid, which means that the protein is not affected. You can also have a missense mutation where a new codon is formed and one wrong amino acid is formed because of that. And then you have a nonsense mutation where the new codon formed is actually a stop codon, which means that the protein is, or the polypeptide chain is ended abruptly and too early. So all the other amino acids afterwards don't get to be formed. And then mutations, there's a whole lot of different results. Obviously, there can be no effect because of it can be a sense mutation. But because there's a change in DNA, there can be a change in mRNA. Because there's a change in mRNA, so a change in the codons, that means that there would be a different tRNA because it's a different anticodon. And if there's a change in the tRNA, it could mean a different amino acid depending on whether that codon is, those two codons are redundant of each other. So the different amino acids could mean that a protein is formed differently, might not perform its structure or its function properly, it might perform a different function. That depends on the mutation. So some mutations, especially point mutations, are insignificant. Very often they're insignificant, but sometimes they have quite a big effect. For example, cystic fibrosis is actually caused by one wrong base. Also, sickle cell anemia is formed when there's only one incorrect base formed. Mutations can be passed on to future generations, but only if the mutations occur within a gamete, so a sperm or an egg cell. But if it's in a somatic cell, so a body cell that's not passed on to future generations, then this mutation isn't hereditary. So mutations can be beneficial, but in humans, mutations aren't often beneficial. If we look at like evolution and stuff, that's where we'd focus a lot on beneficial mutations that increase survival rates. So, for example, antibiotic resistance in bacteria. If one bacteria has a mutation that allows it to be resistant to antibiotics, it's obviously going to survive the antibiotics and be allowed to reproduce and rapidly reproduce, and that's how many bacteria get resistance very quickly. Mutations can obviously also be harmful, such as what we spoke about, cystic fibrosis, hemophilia, Down syndrome. There's loads of harmful mutations. And there can also be neutral mutations, such as different eye colors or blood types came from mutations.